really good set of names for proteins and then give them new names just because we're immunologists. It's quite unnecessary, I think. I try to avoid it.
So most of them do have other names.
They all have other names. They were all things before. Yeah. Very few of them are only
It's just cluster of differentiation, some number, and it gets confusing.
Yeah.
Yeah, there's a lot. Okay, so we are streaming live on YouTube. So our default is to leave your talks up as part of our archive, but we can take them down after a short time or take them down immediately or anything that you feel comfortable with. So you can just let us know your preference on that.
Okay.
But
yes.
If you're happy to leave them up, then lots of people come back and watch them later, or even
for me, for some
later.
time it's fine. I mean, yes, it's fine.
Yeah, I'm fine as well.
Awesome, sounds good. All right, so we always let people keep coming in as the hour starts, and Ankita, you can choose when to start.
I'll just wait maybe like 30 seconds more while I pull up everything.
Great. Yeah, and so I think each of you are prepared to speak for like 22 minutes or something like that to leave time for transitions and questions. And
yes.
Awesome. All right.
Yes, mine is 10:21, but it's never perfect. So maybe it's slightly more or less, but around 21, yes.
Awesome.
Sounds good. Okay, I think we should start now. Sorry, go. You were saying something?
Oh, I was just going to say mine's a little long, but I think I'll leave out the last little bit. So we'll make it work.
Okay.
Awesome.
Sounds good. I think we will start maybe while people trickle in. Welcome everyone again for another session of Cell Migration Seminar. It's always wonderful to have you all here. Today we have two wonderful stories. First, we have Dr. Emily Mace. Dr. Mace studies human natural killer cell development, particularly with quantitative image analysis and cell biological approaches. And through the identification and study of rare individuals with NK cell deficiencies. They are an assistant professor of pediatric immunology in Department of Pediatrics and a member of Columbia Stem Cell Institute at Columbia University in New York. Today, Dr. Mace will talk about the role of CD56, which is NCAM, in human natural killer cell migration and differentiation. So the floor is yours, Emily. And thank you so much for taking our invitation.
Of course, my pleasure. Thank you for inviting me. Okay, everybody can see my screen?
All good.
In the right mode? Perfect. Okay. So I think I gave a very vague title to start, but as Ankita said, I'm going to be talking today about the role of CD56 in NK cell migration and other things. So my lab studies human natural killer cells, and like all immune cells, these arise from hematopoietic stem cells. They are most closely related to other innate lymphoid cell subsets, but they're functionally aligned with cytotoxic T lymphocytes. And we talk about this functional alignment in the context of an immune synapse. So like T cells, NK cells are able to form immunological synapses with virally infected or tumorigenic cells, and then they kill these through the directed secretion of specialized secretory lysosomes that contain perforin and granzyme. So we can think about this as like a specialized cell-cell contact that's this organized signaling platform that leads to the polarization of the NK cell and then the subsequent exocytosis of these lytic granules and killing of the target cell. And so this is kind of one of the best characterized like specialized cell-cell contacts between immune cells. And this is really where I did a lot of work in my PhD and postdoc and thinking about how immune cells interact with each other. But we've become interested in NK cell development. And part of the reason for this is unlike T cells or B cells, there's really sort of very little known about the contact dependent signals that drive NK cell differentiation. So we know that NK cells arise from the bone marrow, and then these early precursors are thought to leave the bone marrow and traffic to secondary lymphoid tissue where they undergo differentiation through sort of increasingly restricted stages of lineage commitment. Then they become mature NK cells and they enter circulation and circulate through the blood and then can be recruited back into tissue. And when we talk about maturation in this sense, a lot of the time we're talking about functional maturation. So the acquisition of these lytic granules that contain perforin and granzyme and the ability to kill target cells. And human NK cells in peripheral blood are defined as being CD56 positive, and they can be further broken down to being CD56 bright or dim, and CD3 negative. So they're different from, for example, NKT cells. Now we can recapitulate this whole process of NK cell development in vitro, And quite often when we do this, what we do is we take CD34 positive precursors from either cord blood or peripheral blood or bone marrow, and you co-culture these with these stromal cells. And this process takes about 4 weeks, but these cells go through all these stages of differentiation and they become these functionally mature NK cells. And so we were interested in this process in the context of additional sort of special cell-cell contacts that could be, in this case, driving NK cell differentiation, because we know that this process is dependent on the presence of stromal cells, and it's dependent on the contact between the NK cells and the stromal cells. So we set out to study this process, and we did this through setting up these co-culture systems where, again, we take these CD34 positive precursors, we co-culture them with OP9 or ELO81D2 stromal cells in the presence of cytokines. And then because we were interested in the cell-cell contacts, we do this using live cell imaging, so long-term live cell imaging. And then we can also do confocal microscopy for fixed cell imaging of NK cells in contact with the stromal cells. And then we can pull these cells off at multiple stages through differentiation and identify sort of the stage of differentiation they're at. And we can also do that in order to align them with cells that we freshly isolate from the tissue that supports NK cell development. And what we've used largely as a model for secondary lymphoid tissue is tonsils, because this is a site of NK cell development in people that can be fairly readily accessed. So, again, we really set out looking for this immune synapse, right? So this developmental synapse, which I naively thought might be this beautiful, stable, long-term contact formed between these developing cells and these stromal cells. But what you can appreciate even from looking at this movie is that in fact what we found was that these cells rapidly became very dynamic on the stromal cells. And so this is an older movie now, and this has been published, so I'm not going to spend too much time on it. But essentially what we showed was at the beginning of these differentiation cultures, the cells were largely non-motile and sort of round. And then when we look back into the same well, in this case, it's 21 days later, these cells have polarized, they're very migratory, they're interacting with the stromal cells, they're interacting with each other. And what we did to characterize this was show that in addition to the acquisition of sort of track length and velocity that occurs through the process of differentiation, their migratory phenotype also becomes more complex. So the cells undergo more periods of or more switches between modes of migration, and we classified those modes of migration as either being directed, undirected, or sort of Brownian or arrest. And so we see that these cells undergo more complex switching between modes as they undergo development. And again, in each case, we also correlated this to the stages of development that we found from freshly isolated cells from tissue. So we went on to show that this is mediated by cell adhesion molecules, and it's also partially mediated by extracellular matrix components that are secreted by the stromal cells, so are acting as an adhesive layer for the NK cells. And that, again, as these cells underwent differentiation, this sort of the spontaneous migration was associated with their development. Now, one aspect of this that we were particularly interested in was this behavior that we noticed that was describing one of their types of arrest behavior. And this was this tethering through this sort of protrusion that was coming from the NK cells. And so we can see an example of this shown here on the left. We can see that this is marked by the polarization of CD56, which is one of our markers of interest. And we can see that this cell is really seemingly adhered to these stromal cells with high affinity and undergoing almost sort of 360 degrees of rotation around this point of tethering. And we see that this is specific to adhesion to the stromal cells. When we put these cells on just an antibody-coated surface, they aren't intrinsically polarized. When we look in the Z-plane and we look sort of sideways into the cell, we can see them tethering through this protrusion. And although it's not very clearly shown in this image, what we also marked this by was polarization of the centrosome sort of down into the neck of the cell. And then finally, we also showed that this was a site of signaling, so we could mark calcium flux and tyrosine phosphorylation that happens at this site. So really suggesting that there's this cell tether that's this dynamic structure that may also be a site of signaling in these cells that are undergoing arrest on these stromal cells. So we aren't the first people to have described this, and when we look back in the literature, we actually found that Max Krummel described something similar 15 years ago. And in this case, it was in the context of a T cell interacting with an antigen presenting cell, or an APC. And what they described were these tethers that they think are sort of surface-bound chemokines that are priming the T cell for further interactions with the antigen presenting cell. So you can see an example of this in the top panel here, where there's this tether that forms with the APC. And then you actually see that what the distal side of that cell then wraps around and forms this immune synapse with the antigen presenting cell. And so what they were suggesting is that, in this case, this tether was this initial point of contact mediated by chemokines that then sensitized the cell for immune synapse formation after that. And then more recently, we've seen a similar thing published from Manuel Terri's group, showing similar interactions, in this case between hematopoietic stem cells and osteoblasts, which help promote HSC survival. And again, we see a very similar structure. When we're looking in the Z plane into the cells, we see this tethering through this sort of protrusion. And again, this is marked by polarization of the centrosome, as well as the polarization of adhesion molecules and chemokine receptors, including CXCR4. So again, suggesting that this may be a sort of a conserved structure which has some form of function. Now, I would be the first to suggest that this may be sort of almost an artifact of some of these sort of two-dimensional culture systems that are done in the absence of shear flow. So we haven't applied shear flow to this system, so we don't know if that's going to change these, but these are certainly conserved in these sort of 2D static cultures. And one of the reasons I bring this up is quite often when I mention these, people go, oh yeah, we see this all the time and we don't know what they mean. So we're kind of interested in them as a site of signaling. Now we think that this structure is uropod derived, and we think that because we actually see it forming from the uropods of cells that are migrating. So this is an example shown here on the left of an NK cell that's migrating and then seems to tether through its uropod and then undergo this sort of high affinity tethering and rotation around that point of contact. And similarly, when we look in fixed cell microscopy, we can see the polarization of uropod associated markers, things like Mace and CD43, at the point of contact with the stromal cells, again, suggesting that it may be a uropod-derived structure. Although there has certainly been some debate about that, and I think, in particular, Manuel Terri's group may not think the same thing. So, but we think that this may be playing a role, in terms of function, it could be playing a role in establishing polarity. Particularly, we see the polarization of the centrosome down into the neck of the cell. And again, we see the accumulation of many of these signaling molecules, including Notch and CXCR4 and beta-1 integrins. So we think this could be setting up a context for asymmetric cell division, or as in the case of the structure that Max Krummel described, acting as sort of a priming step prior to additional interactions that occur. And so we haven't gone too far down that road yet, but one of the things that we have done is start to look at different integrin ligands, and what we see is that the beta-1 integrin ligands, so in particular fibronectin and VCAM, tend to promote this tethering more than beta-7 integrin ligands, and both of those more so than beta-2 integrin ligands. And all of these are integrins that are expressed on NK cells. And this perhaps isn't too surprising given the known association of beta-1 integrins with the midbody and the cell uropod, whereas beta-2 is more associated with the leading edge in immune synapse formation. But what I want to talk about for the second part of the talk is the role that CD56 plays in all of this. And so CD56, of course, is, aside from being a CD marker, is NCAM. So it's neural cell adhesion molecule. It's expressed on all human NK cells, and I emphasize that because this is one of the reasons why it's been kind of poorly studied in the immune system, is it does seem to be specific to humans and non-human primates, but it's not found on mouse NK cells. And NCAM is a member of the Ig superfamily, so it's got 5 Ig domains, 2 fibronectin domains, and then it's also special because it can be modified by the addition of this polysialic acid. So it's glycosylated, and then you see these large linear chains of polysialic acid, which give it this really large, sort of bulky, negatively charged residue on the cell surface. NCAM can be expressed in 3 isoforms, and we and other people have shown that the primary isoform that's expressed in NK cells is NCAM 140. And the difference between these is that, of the 3 isoforms, NCAM 120 is GPI linked, whereas NCAM 140 and 180, and this refers to their molecular weights, contain a transmembrane domain as well as an intercellular cytoplasmic domain that can mediate signaling. And so it's been really poorly characterized in NK cells. What we know from studying it in neurons and in the nervous system is that its role can be in adhesion. So it can mediate adhesion by binding to other NCAM molecules, both in cis and in trans. And this is further affected by the addition of polysialic acid. So the removal of polysialic acid actually strengthens adhesion because you remove those bulky negatively charged residues. It can also signal by binding either directly or indirectly to focal adhesion kinase and the Src family kinase FIN, and it promotes neurite outgrowth, which is essentially actin remodeling and wayfinding. And then it can also, particularly the NCAM 140 isoform, can move in and out of lipid rafts and help to recruit signaling molecules that way. And so, we didn't there's been a little bit of work done on sort of the role of NCAM in NK cells, but its function was very poorly understood. And this was work that was begun in the lab by Amara Martinez, who was actually an undergrad, who's now gone off to grad school, and now is being partially continued by Michael Shannon, who's a postdoc in the lab. So the first thing that we did was we knocked out CD56 on 2 human NK cell lines, so NK92 and YTS cells. And what we found really surprisingly was that it actually inhibited cytotoxic function. And so we can see this when we look at the graph on the left here, which is a graph of these NK92 cells killing K562 target cells, where we see decreased function of the CD56 knockout line, which can be rescued by the re-expression of full-length CD56. And so we delved into this a little bit more deeply, and what we found was there was this sort of overall repression of phospho-PIK2 signaling. And again, we can see this quantified here, where we see that the CD56 knockout cells have decreased global phosphorylation of PIK2, which can again be rescued by the re-expression of full-length CD56. So this was kind of interesting to us and not unsurprising, given that PIK2 is the hematopoietic homolog of focal adhesion kinase. So given the link between FAC and NCAM that was shown previously, we were not too surprised to see that there was sort of this defect. And of course, PIK2 functions downstream of integrins in order to promote actin remodeling, and has been previously shown to play a role in NK cell cytotoxicity by promoting the polarization of the cell towards the target cell. And so indeed, that's what we found when we look at these conjugates shown here on the left. This is a CD56 knockout cell. So we see decreased PIK2 phosphorylation at 2 different tyrosine residues. And this is accompanied by the decreased sort of polarization of all that lytic machinery towards the target cell, and accompanied by decreased exocytosis, target cell killing, and cytokine secretion. So that was kind of interesting, but we didn't really delve too deeply in this study into sort of what the actual mechanism was. And so we wanted to look more closely at what CD56 was doing in cell migration in particular. So the first thing we did was look at its localization, and what we found, really strikingly was that we seem to have 2 pools of CD56, and this is an NK cell spontaneously migrating on ICAM that's been adhered to glass, and we're looking down at the cell. And what we can very clearly see is that there's this pool of CD56 that's found in the uropod. We find it co-localized. In this case, I'm showing CD44, but we've also done this again with phospho-ERM molecules and other uropod-associated markers. And we can very clearly see this sort of bright punctate pool of CD56 that's found in the uropod. But we also see a second pool that, although it doesn't look as strongly expressed, is co-localized with actin, and we can very clearly see that here as we see, the expression of CD56 in the lamellipodia and in other actin-rich structures in the cell. And we can perhaps see it a little more clearly in terms of the uropod localization when we actually look sideways into the cell. Lymphocytes are kind of notorious for elevating their uropods as they migrate in order to go faster. And so we can actually see that you see the elevation of the uropod, the direction of the cell movement is this way. And we see the co-localization both of CD44 and of CD56 in the uropod. So this was interesting but we were particularly interested sort of in this actin co-localization as well. And another way that we looked at this, which I think is also really striking, is to go back into our images of NK cells conjugated to target cells. And here again, we see that as this NK cell is formed a conjugate with the target cell, and we can almost see this. Yeah, of course.
It looks like Golgi. Is it Golgi?
Yeah, that's a good question. We haven't done co-staining with the Golgi.
Because the Golgi's back there, right? And it looks pretty stacky to me.
Yeah, and it's not.
So it could be getting sialylated. It could be in the process of getting sialylated before it gets secreted.
Yeah, that's a great idea, and that makes a lot of sense, and that's something we can very easily look at. So thank you. And so when we look back into these conjugates and look at where CD56 is here, it's really striking because we see this sort of cap at what is now the distal pole from the immune synapse, which I associate with the uropod because we know that cells form an immune synapse with their leading edge. And then we also see this sort of this co-localization with actin, and we can see it quite clearly when we then take the immune synapse and rotate it and look back up into the cell, and we can see this very sort of canonical ring of actin that you see at the immune synapse that's also shadowed by this ring of CD56. So we don't know what the significance of the 2 pools is, whether this is polysialylated and unsialylated or whether this is different isoforms that might be expressed, but it's very clear that there's sort of these 2 localizations that are present within these cells. So we then went on to look at cell migration, and this is just using confocal microscopy, in this case 20x. And we've got the wild type cells on the top panel and the CD56 knockout cells on the bottom panel. And even before I play the movies, you can very sort of qualitatively see that there's differences between the cells. And again, this is on iCam that's fixed to glass. And what we can see is that in the wild type cells, you see the cells migrating, obviously. In the CD56 knockout cells, they've got this very distinct phenotype where they look very irregularly shaped. And then when we play the movies, we can see that although the cells are somewhat dynamic, they aren't migrating as much as the wild type cells. And then to quantify this, we're performing segmentation using cell pose and tracking using Bayesian tracker, which is developed in Alan Lowe's lab. And again, this is an analysis pipeline that's being developed by Michael Shannon in the lab. And again, before I even play the videos, you can appreciate the different shapes that are identified by the segmentation. And one of the strengths of using this approach is that we then get the shape parameters in addition to the information about the cell tracks as well. And this is a bit disco because these are colored by frame, so I apologize for that. But again, you can appreciate just looking at the segmentation that the CD56 knockout cells are this very sort of distinctive phenotype. And although they're dynamic, they're not migrating as much. You can see this in another way if we look at the cell tracks from first the wild type and then the CD56 knockout. And when we look at the accumulation of tracks at the end, we see that the CD56 knockout tracks are shorter. And so to quantify this, we've plotted the effect size to sort of avoid overpowering our statistics. And what we can see is, these cells migrate at sort of a predicted speed for lymphocytes, so about 10 microns per minute. And we see a significantly decreased speed of migration in the CD56 knockout cells. And then again, as indicated by the segmentation, we see this increase in their eccentricity parameter as well as in the cell perimeter. So they've got this very sort of distinctive phenotype, which we can identify even at 20x. When we look more closely, in this case, it's using structured illumination microscopy, we can see a very distinct dysregulated actin phenotype. So when we look at the wild type cell on the left, again, we see this sort of broad lamellipodia, clear direction of motion with a trailing neuropod. Whereas in the CD56 knockout cells, we can really identify what we've thought of as three key features. So one is that there's some dysregulation of the lamellipodia. Two, there are these large actin foci which form in the cell body. And then three, there are these elongated and sort of spiky, unhappy looking filopodia. Now, because, and you may have noticed, these are cells plated on fibronectin. The previous slide was cells plated on iChem. And so we wanted to go back and sort of ask how conserved this actin phenotype was. And to do that, we've done something that we've characterized quite extensively, which was we've plated these cells, again, by cross-linking LFA1 and an activating receptor to kind of recapitulate and now a symmetrical actin spreading on glass. So in this case, the cells aren't polarized because they're not migrating, but they form this symmetrical spreading. And again, we can identify these same features that we saw in the migrating cells in these symmetrically spread cells. So again, we see these large actin foci in the CD56 knockout cells and these long filopodia. Now, importantly, when we put back in the full-length CD56, in this case, we're using NCAM 140, this rescues the phenotype, which is shown here on the left. And when we put back in a construct that's missing the intracellular domain, so it contains the transmembrane, but not the intracellular domain, it does not rescue. And this is quantified on the right here. So really showing that this actin phenotype, which seems to be involved in sort of modulating actin homeostasis or actin remodeling under different integrin ligand bindings, it requires the intracellular domain of NCAM. And this is just to show that when we express these constructs, we detect CD56 on the cell surface at a lower molecular weight when we delete the intracellular domain. And we further went on to show that these actin foci that we're seeing are ARP23 dependent. When we treat the cells with CK666, then we see that we lose both in the wild type and the knockout, we lose these large actin foci. So we initially thought this may have to do with sort of actin remodeling. When we look more closely at these cells migrating, in this case, this is at 100x biconfocal. In the wild type cells, again, we see this sort of very characteristic ruffling at the front end and then sort of canonical cell migration. The CD56 knockout cells are actually still quite dynamic. So we see the actin ruffling, but we really see that the cells fail to polarize and migrate. And so we instead started to focus more on sort of the idea of detachment, and maybe these cells are unable to detach integrins and undergo migration that way. And this was supported by looking at using monoclonal antibodies to look at activated integrin or open conformation integrins, where we found that on the wild type cells, When we look at the CD56 knockout cells, they have increased presence of open conformation of, in this case, beta-2 integrin, and this is played on using LFA1 ligation. So we're ligating LFA1. We see more activated integrins, again, suggesting it may have something to do with with integrin detachment or turnover. But what was also surprising to us was that when we ligated beta-2, we saw a similar thing happening with beta-1. So we're seeing increased open conformation of beta-1 integrins, even when we put the cells on beta-2 substrates, essentially. So then we started thinking about whether this has to do with sort of integrin crosstalk, or perhaps whether it has anything to do with the surface that we're plating the cells on, and maybe these cells are just undergoing some sort of deficient actin homeostasis at baseline. And that is what we actually see. So when we take these cells just out of the cell culture flask, and we put them on polyol-lysine, which is a fairly inert surface, we still see the presence of these sort of dysregulated actin filopodia. And similarly, when we put these cells on polyol-lysine and image at the plane of the glass, again, we see that the filopodia and the large actin foci are present, even in the absence of any sort of integrin ligation or integrin cross-linking. So really just suggesting that this is happening at baseline. So in the interest of time, I'm going to skip over sort of some of the mechanistic data. I'm going to summarize it by saying that we looked more closely at the PIC2, and we found that although overall phosphorylation is decreased, we actually find it enriched in those actin foci. Similarly with vinculin, so suggesting that these proteins are kind of being trapped in these actin foci that aren't undergoing sort of normal homeostasis or turnover. And lastly, what we've done is we've done phosphoproteomic analysis, where again, we've just taken these cells out of the flask and looked at the phosphoproteome using 2 different NK cell lines, wild type and CD56 knockout. And what we see are the biggest differences are, not surprisingly, in cytoskeletal organization, as well as in the regulation of GTPase activity. And so we've done some initial looking at CDC42 in particular, and this is where I think Claire's question is also very relevant, because we also know that CDC42 can be associated with a Golgi, so that rings a bell. And we see this that there may be this dysregulation in CDC42 GTP binding in these cells as well. So what do we think is happening? The way I'm thinking about it is a model where we've got things happening on the inside and on the outside of the cell. So one of our hypotheses is that NCAM may be acting as a sort of parking lot for all these proteins that are involved in actin homeostasis. So it's acting to hold them there and have them then be available as they're required for actin turnover and remodeling. But then I think it's important to not, it's hard to ignore sort of the presence of these PSA molecules that are large and negatively charged on the surface of the cell, which is unusual for cells. We know that it's kind of special to immune cells. And whether that's playing a role in modulating the spatial localization of cell surface receptors, and then potentially the recruitment in and out of lipid rafts as well, is something that we're very interested in. So in the end, what I've told you is that it actually doesn't have a lot to do with cell migration. It has to do with a lot of things, but ultimately what we're interested in is cell migration. We're particularly interested in this uropod localization, and also the role of the uropod in these specialized tethering structures that we see. So with that, I will end. I've tried to sort of thank everybody as we went along, but a lot of this work was done by Amara Martinez, who's now doing her PhD at the NIH, and then Michael Shannon, who's a postdoc in the lab. And happy to take questions.
Thank you so much, Emily. That was wonderful talk. While we wait for a few questions, I actually had a question, and you sort of talked about it in the end about the uropod thing. So because to me, when they were migrating, it looks like they are able to form some lamellipodia. So it really looks like that uropod is the issue. But did you have a chance to look at anything like Ezrin or CD44 to whether it's still localized somewhere specifically, or is it more uniform?
Yeah, we've looked a lot at that, right? And so, and I didn't include the CD44 data because it was a little hard to know how to quantify it. So we certainly, it's not so clear as to say that it doesn't form, it seems to still form a cap somewhere on those cells.
Yeah.
So when we look at the CD56 knockout cells, you can see sort of this patch of CD44, but it's hard if the cell hasn't polarized to know whether that's a uropod or whether that's just sort of a patch somewhere. So one of the things that we're, I've sort of been thinking about is how do we quantify the mislocalization of some of these proteins and what.
So how about when they are actually attacking those, I forgot the other cell, the 72 one, like when they actually form the synapse, because that needs a specific polarized mode, right?
Yeah, that's a good point. We should look at the CD44 there and see if it's still sort of distally localized. Yeah.
Cool. I think we have questions, right? Okay. The first question is from Diaz Bello asking, sorry if I missed, the purpose of co-culture mentioned at the beginning. Is it for the development or the maintenance of natural killer cells? And which type of extracellular matrix is usually used by NK cells in order to get polarized morphology and migrate?
Yeah, that's a great question, because those stromal cells can be used also for the maintenance of HSC cultures. But in our case, they promote differentiation. But again, it should be noted that's with the addition of cytokines as well. And we didn't really extensively, we didn't do the proteome analysis on the extracellular matrix that's secreted. We do certainly find collagen, laminin, and fibronectin in there.
Awesome.
Okay, great. We have a question from Haroon asking if you checked the distributions of integrins and the knockouts, and also is there a difference in myosin phosphorylation upon knockout?
Yeah, both great questions. We have looked at the distribution of integrins, and again, because the cells don't polarize so much, it's hard to sort of assign locations to them. What I can say is that the overall expression of total integrin is not affected. But aside from doing the activated, we haven't really looked closely at the. It seems like they still kind of go where they should. So you do see beta 2 sort of towards the cell membrane, and beta 1 more in the mid body, but yeah, a little hard to say.
Fantastic. Next question is from Sangyun saying, very nice presentation. How would you relate the uropod to the localization of actin foci?
Yeah, that's a good question too. I think the actin foci really seem to be on the ventral part of the cell that's touching the surface. So one of the things that I've tried to do is go back and look and ask whether, are the actin foci on the top of the cell as well? Because if we're really saying that it's sort of to do with this cortical actin homeostasis, then it should be also all over the cell, but it's hard. It's hard with sort of 3D confocal to really get the resolution to convince ourselves of that. But looking at the cells on polylysine, it really does seem to be where the cell makes contact, and sort of in the mid body of the cell, which again may reflect some interaction with beta 1 integrins too.
Great, thank you. We have a couple questions from Si Chang, saying that CD56 is a cell-cell adhesion molecule, but you're showing behavior of single cells. So is the CD56 engagement required for regulation of cell behavior? And then the second question, is it known what is the receptor of PSA?
Yeah, so we looked at just sort of adhesion, and we didn't find defects in just cell-cell adhesion. There's been a lot of conflicting data on whether it mediates interaction between NK cells and other target cells, and it depends on the target cell you use. So people have looked at sort of CD56 or NCAM expressing targets or NCAM negative targets, and there's conflicting data on that. So I don't think there's a big role for it in mediating NCAM adhesion, although it does seem to accumulate at the point of contact between NK cells when they're talking to each other. But based on our experience, I think that it is not necessarily doing that.
Okay, awesome. So I think we're actually perfectly on time, which is really surprising for us. There's one more question in the chat, Emily, but I'll let you maybe answer that so that we can move on to our second speaker. So I will go ahead and introduce you, Esteban, and you can start your screen share in the meantime. So we are delighted to welcome Dr. Esteban Hoijman. I will first apologize that I misspelled Esteban's name in the mailers. So if you are going to go and cite his work after the amazing talk he's about to give, search for his name the way he has it spelled on his screen, not the way I spelled it in the email. So apologies there. All right, so Esteban obtained his PhD at the University of Buenos Aires in Argentina. He went on to do postdoctoral research at the University of Pompeu Fabra and the Center for Genomic Regulation in Spain. And then he also did short postdoctoral stays at the Hubrecht Institute in the Netherlands, King's College London in the UK and the University of Toulouse. So he's really been all over the place, which is really awesome. And then in 2021, he began as a lecturer at the University of Barcelona and also as a group leader at the Institute of Biomedical Sciences at Bellvitge in Spain. And so, of course, we always give mad props to anyone who started their own group in the middle of the pandemic. And so we are really excited to hear today about work that he's done involving quantitative imaging of dynamic processes across scales in live embryos. So Esteban, take it away. The floor is yours. Thanks so much.
Yes, thank you very much, Jennifer, and the organizers for inviting me to talk. And I think that is a very nice seminar series, and I really thanks to the opportunity to talk. So yes, as Jennifer said, last year I started my lab, and what we are interested in the lab is trying to understand embryonic development, but in addition to most of the labs that study embryonic development and cell dynamics, that they understand cell dynamics from the genetic point of view, and there's a lot of single cell sequencing going on now. We have to also understand the dynamics of the cells in terms of movement, shape changes, and this type of features. And we want to focus not only on what the single cells do as the right cells that you are seeing now, which is a stem cell in the interior of a zebrafish embryo, but also how they interact collectively as the left image. So for this, we use a quantitative microscopy approach to understand the cell dynamics and also the mechanics of these processes. Today, I will tell you about this story that we published last year and was part of my previous postdoc. And I want to start by showing this movie in which we will see the development of a zebrafish embryo, and the right embryo is a mutant. It doesn't matter the mutation, but it's clearly failing in development. But I want that we focus on the left embryo, and the left embryo is apparently developing normal. And the question is developing normally because nothing was going wrong, or maybe there were some mistakes going on and the embryo was able to deal with these problems. So what kind of problems an embryo can face, from environmental stressors to internal problems like mechanical tasks that the embryo has to do. And in fact, it is known that human embryos do have these errors because most of the embryos are mosaic in chromosome number due to errors in mitosis. And it's believed that this type of errors, in this case, the error in mitosis, are the main cause of failures in human preimplantation development. So the question that we wanted to arise is how early embryos deal with an anomaly. If we think in an adult organism, we have the immune system to remove, in case that we have defective cells, using this tandem of apoptosis and phagocytosis, in which the defective cells die by apoptosis. This is recognized by a professional phagocyte of the immune system and remove this cell by ingestion. But in the early embryos, we don't have professional phagocytes, so we can shift the question to try to answer how to remove apoptotic cells in absence of phagocytic cells. So if we analyze the architecture of early embryos and we compare different vertebrates as mammals and our main experimental system, that is the zebrafish embryo, we notice that we have two types of cells. Mainly, we have the first tissue that is formed, the first functional tissue, which is an epithelium that is forming the surface of the embryo, that in mammals is called trophoblast and in zebrafish's enveloping layer, or EBL. And in the interior, we have the progenitor stem cells that will form the embryo, that is conserved in different vertebrates. So when we wanted to understand how the dynamics of these cells are occurring in the interior of the embryo, and if we can capture some of the errors that are taking place in the interior of the embryo, we filmed these early zebrafish stage embryos in blastula and gastrula stages. And what we observe is what you will observe as an example in this cell. This cell is dividing, and then we have two daughter cells, but an intercellular bridge is remaining there, which finally results in a fusion, and we have binucleated cells. These binucleated cells are occurring spontaneously at different frequencies in early stage embryos. And what happened with these binucleated cells is that these binucleated cells, when they are going to divide again, they make sometimes strange divisions, like tripolar divisions, and the three daughter cells, they die, and we can mark these cells and identify that these cells are dying by apoptosis. What happens with these apoptotic cells, and this movie is slightly more complex because it's shifting in set during the movie, because at the beginning you are seeing the internal cell, the stem cells, but at the end you will see the surface of the embryo, the epithelial cells. And this is what is going on. That a cell in the center of the image is changing shape, is losing shape, is fragmenting. And when we track in 3D and in time these fragments, we observe that these fragments end in the surface of the embryo, now in the interior of these larger cells that we can see now, that are the epithelial cells of the surface of the embryo. So a process of ingestion took place during these frames. This is more or less what you see, is that a cell loses shape, fragments, and the epithelial cells are clearing these fragments by ingestion. And next, we wanted to know if this process that we observe that happens with the spontaneous cells that are dying, what happens if the embryo has a lot of cells dying? How efficient is this system to clear dead cells in the embryo? So for this, we manipulate the system now, and we challenge the embryo by creating a mosaic embryo that overexpress apoptotic protein bags in the interior of the embryo, so the red cells that you are seeing here. And when we do this, if we use high levels of bags protein, this is a survival curve, and the brown curve is what you can see here, is that the embryos die very soon, but an intermediate level or even low levels, we see different responses. And for example, the pink curve, which has relatively low level of bags, the embryos are able to deal with this problem and survive perfectly. And what is going on in these embryos that are surviving? So in the interior of the embryo, we can see that in fact we have dead cells. We have around 100 of dead cells. And in this case, which are the red cells that you can see here in the interior of the embryo, and in the upper image is the surface of the embryo. And if we analyze the dynamics, and you see in the upper image, you start to see that these red dots start to appear in the interior of the epithelial cells. So a process of ingestion is taking place. And after 2 hours of this process occurring, in the interior of the embryo, we have kind of depleted the dead cells, and in the surface of the embryo, these epithelial cells were ingesting most of these cells. And this is what is quantifying here, that in the black curve, you can see how the internal content of dead cells is being depleted, and the surface start to acquire these fragments by ingestion. And this is a general stress response, because it's not only happening when we overexpress Bax protein, but if we damage the DNA by irradiation or induce mitotic errors by inducing the overexpression of topoisomerase that are defective, or induce mitochondrial stress, different type of stress, we always observe that an apoptotic cell is present in the embryo, is cleared by this system of ingestion by epithelial cells. And to understand how this ingestion was taking place, we thought this should be a phagocytic process, but we wanted to analyze this in more detail. If we have a phagocytic cap taking place, we should see these protrusions that are filled with actin that are forming these phagocytic caps of the professional phagocytes when they are phagocyting. And to visualize this in the embryo, we stained single cells of the epithelium to visualize better the process. And in cyan, you can see a live actin staining, and interior you are seeing in red the dead cells. So in this image, in which you can see in the upper image, a section that is parallel to the surface, or in the lower image, a section that is transverse to the surface, we can see this process with more detail. And you can see that in fact it's a phagocytic process, that we can see the protrusions that are engulfing this particle and include actin enrichments in these cases. So we have a functional phagocytic cap that we can visualize even in 3D with and obtain more information about how this process is going on. Here we have this single cell, epithelial cell, that is trying to ingest this apoptotic particle, and during ingestion in these protrusions, what is forming in fact is an actin ring that is surrounding the apoptotic particle, and in fact is compressing this particle to propel the particle to the interior of the embryo and execute the ingestion itself. This is what is shown here in this quantification of the curvature of the apoptotic particle, and you can see that there is a compression going on. So it's a mechanical compression, and we are now studying how is the mechanics of this phagocytic process that is taking place in epithelial cells in live embryos. And then we checked that this process is a phagocytic process from the signaling point of view, because the typical signals that participate of phagocytosis, like RAG1 or PA3 kinase, are participating of this process. And then after phagocytosis takes place, the phagosomes are functional. And in fact, the content of the phagosome is being degraded. Next, we wanted to know how the apoptotic cells are recognized, and usually the apoptotic cells use different signals. One of these signals is phosphatidylserine, this lipid that is exposed from the external side of apoptotic cells at the professional phagocytes are able to recognize. And in this case, we observed kind of the same process, and we did several experiments to prove that it's the same mechanism that in professional phagocytes. But in this case, I am showing this experiment, which is interesting for several reasons, in which we introduced inside the embryo these lipid aggregates that when they contain this phosphatidylserine, this is what is going on. They are in the interior of the embryo. When we analyze what happens is that the epithelial cells are able to recognize the presence of this phosphatidylserine and to ingest these lipid aggregates. And this only happens when phosphatidylserine is present. If phosphatidylserine is absent, this is not taking place. And this experiment is allowed us to do another interesting experiment, which is try to use a toxic level of these lipid aggregates. If we inject enough amount of lipid aggregates, the embryos start to die. But when they, with these aggregates include the phosphatidylserine, they die less. So in some way, when they can clear these lipid aggregates, they survive better, supporting the idea that the clearance that we are observing is important for embryo survival. And next, we wanted to know if this process is only happening in embryos of zebrafish or this is conserved in mammals. So we did several experiments to prove that in the mouse model is still occurring in a very similar way. So in the mouse model, we have in the interior the inner cell mass that are the stem cells that will form the embryo, and the trophoblast, this surface epithelium, which is similar to the epithelial cells of the zebrafish, that will form the placenta. And when we film this, in this case, are spontaneous events of cell death that we detect, and we check that there are some events of cell death that are taking place, marking apoptosis marker, and these cells are released to the cavity, and after the release, they are ingested by the trophoblast. Okay, and this is happening in a very consistent way, and the number of cells that are dying spontaneously is very high in mammals, and is also happening when we did another experiments, like is injecting apoptotic cells in the cavity that they are cleared by the trophoblast, or when we use these phosphatidylcoated targets, they are also cleared by the trophoblast when they have this phosphatidylserine, and they are not detected when they do not have. So for this part, we can conclude that the vertebrate embryonic surface epithelium works as an efficient apoptotic clearance system that removes defective cells. In some way, these epithelial cells acquire some type of immune roles. It's just a phagocytic detection of apoptotic cell. This is something that the immune system does to protect the embryo in this case. And this system allowed us to try to tackle another question, which is something that is curious about how epithelial cells can clear apoptotic cells, which is something that happens also in adult systems like in the lungs or in the intestine. So in this case, what we are seeing is just macrophages and neutrophils working together to be recruited to this site of damage that they have to heal. And as you can detect, and you know already, these cells are very motile and they act collectively and they can change shape a lot. But the epithelial cells are non-motile phagocytes that they have restriction also to change shape because they are attached to the neighbors. So the question is, how can they perform efficient clearance? And what we notice is something very curious that is interesting from the motility point of view too, that for the specialists in cell motility, that is that when we film these cells before they are ingested, what we observe is that they acquire a very fast motility with random directionality, okay? And this is very different to the motility that the live cells has here. The red cells are live, healthy cells, which are moving very slow and directionally as part of the gastrulation movement of the embryo at this stage. If we compare the motility in terms of directionality of the two populations, we can see that it's very different, and also in terms of speed. So the apoptotic cells are much faster than the live cells, which is curious. So to understand better this motility, what we analyze is this with more closely, and what we observe is that when the cells, apoptotic cells, are moving, they are moving in association with these accumulations of actin in cyan, here that we can visualize here, and these actin accumulations are not in the interior of the apoptotic cell, but they are coming from the outside. You can see this better here in this high-speed movie, and you can see that when the apoptotic particle is moving, there are actin filaments in contact with the rear part of the apoptotic particle, not in the interior of the apoptotic particle, from the exterior. And this is something that we quantify when we quantify the angle of movement and where the actin accumulations were taking place and what's in contact with the rear surface. And to detect from which cells this actin was coming from, we again use this single cell staining. And only when we stained these epithelial cells, this is what we observe, a new type of protrusion that we didn't notice it reported before. That is a very extended protrusion that is moving coordinately with the apoptotic particle. Okay, this is enriched in actin, it's very elongated. We call this protrusion epithelial arm because it seems to be grabbing the particle. And so for the moment, we can say that the epithelial cells can create two types of protrusions, a phagocytic cup for ingestion or an epithelial arm. And we wanted to understand what is the mechanical connection between the epithelial arm and the apoptotic particle. And we did several experiments to test this. And we are presenting here two options. Okay, one is that the epithelial arm is chasing an apoptotic particle that has intrinsic motility. That is something that was proposed for some apoptotic cells in some in vivo analysis of apoptotic cell death. But for us was very conflicting this because the reorganization of the protein content in the interior of the apoptotic cell is very complex to think that this can lead to an organized and migration pattern of these cells. And an alternative explanation is that apoptotic cells do not have intrinsic motility and that the epithelial arms are directly mechanically pushing these objects and propelling and creating this motility that we observe in the apoptotic particles. And we did several experiments that supports that the second hypothesis is the correct one. And I am showing here this one that use again these lipid targets that for sure they don't have intrinsic motility and they move very similar to the apoptotic cells with phases of fast movement and the random directionality, and always in association with a rear accumulation of effect. Okay, so this again supports that, in fact, what the arms are doing is pushing mechanically these apoptotic fragments. So how we visualize this movement is that an arm can push this apoptotic fragment, and then another arm can come, and this second arm can come from a different cell or from the same one, and another one, until we establish an apoptotic path that we observe it. And this is something that you can visualize more or less here, in which you see one arm forming that then disassembles, and then another one that comes from another cell and disassembles, another one, and this can occur consecutively to establish this path that we were observing. And in fact, this is in agreement with the fluctuations in speed that we quantified for these apoptotic particles. And when we fit how they move to different models, we observe that they fit with a persistent random walk, so with phases of directionally and fast movement and intervals of the random movement and slow movement. This is more or less how we visualize it, that they can be traveling before they are ingested. And during this traveling, this phase can be important and can help us to understand why these arms are there. No? So we wanted to address this question about why this is going on. What is happening. And what you can see here is that during this trip, they are facing many epithelial cells, and these epithelial cells are phagocytes. So in some way, this traveling is increasing the encounter rate of these targets and the phagocytes. And this is something that you can also visualize here in this grid that represents every EVL or epithelial cell. And in these circles, you see events of ingestion, and the color code is just the time of ingestion. And if we follow this red cell that is traveling all along this path, until the moment in which is phagocytosis, what we observe is that it's facing all these epithelial cells, okay, during the trip. And when we quantify this, is something very obvious. When they travel, they face a certain number of epithelial cells, a certain number of phagocytes. But when we block the motility, they are facing less number of phagocytes. And then this can be interesting for us in terms of trying to understand that if we have phagocytic cells that cannot move, maybe the movement of the target can help to the clearance in some way. So what happens at the tissue level is that the apoptotic particles are being spread. So in this case, we have the spreading, and if we block the motility, the spreading is much less. And this can be important and can be visualized in this case when we made a transplantation of some apoptotic cells to a localized region of the embryo. And you can see in this movie, and this is represented by these yellow dots, and all the dots that you will see here are real data. And these transplanted apoptotic cells are surrounded by a certain number of epithelial cells that can clear them. But during the process of spreading of this apoptotic group of cells, they now face many more epithelial cells, expanding the epithelial pool and representing a kind of an inversion of what happens with professional phagocytes, that they can move around certain objects that they want to clear. But in this case, it's an inversion in which when the phagocytes cannot move, what is being moved are the targets. And to understand if this has an impact at a tissue level in clearance, what we perform is some computational analysis and mathematical modeling in which we have an unidimensional epithelial layer that has to clear a mass of apoptotic cells and can uptake with some probability or push with some probability. And from here we derived a mathematical formula in which the main conclusion from this analysis is that when we increase the speed of the movement of the particles, what is decreased is the time that is required for clearing the whole apoptotic mass. Okay, that means that increasing the speed of spreading is increasing the efficiency of the tissue. Reducing the time for clearance is increasing the efficiency. But this only happens when we have something that is limiting the uptake, because if this cell, single cell, can uptake everything in 1 second, we should not have this type of behavior. So what we thought is that maybe there is a maximum capacity for these epithelial cells to uptake a certain number of apoptotic particles, as was reported even for macrophages, that they can change the shape a lot, but of course, if they have to clear a lot of target, they have a maximum capacity, because also they have to consume the intracellular proteins for this degradation path that they have to take place in the interior of the cells. So if this is plotted here, that is representing the maximum capacity that each cell has for uptaking particles, and to validate that this has a experimental sense, so what we did are some experiments in which we saturated the embryo with apoptotic targets that we transplanted from other sources. And what we observed is that in fact we can visualize a saturation level in each cell for uptaking, even when there are many more cells that the embryo could clear, each individual cell is being saturated. Okay, and this is supporting the idea that if we share the targets, we share the food, in some way the phagocytes can clear more and the tissue is faster in the clearance process. So what I was showing is that at the cellular level, we have a mechanical pushing that creates an emergent tissue function of spreading the debris, and this can be lead to an increased clearance efficiency. And in this way, these epithelial cells that has to clear and are phagocytes are cooperating mechanically to clear this apoptotic mass. And with this, I finished. This is the cooperative clearance team that the work was mainly done at the Center for Genomic Regulation in the lab of Verena Ruprecht. And the two main people that working in this project, in addition of me, were Hannah Hakkinen and Keral Tolosa. And we also received some support from the sequencing from the Manuel Irimias group and the modeling from Stefan Visser and Andrew Kalanschotz. And as I said at the beginning, I am now starting my lab from 6 months already. And this is my small group, and I am very interested in recruiting postdocs and PhD students. So the people that are interested can contact me and come to Barcelona and to our institute. And yes, thank you for your attention.
Fantastic. Claire, do you want to go first?
Sure. That was amazing. The imaging was spectacular and just amazing to think that that's going on. So, I mean, I might be showing my age, but I never learned in biology that epithelial cells, you keep calling them professional phagocytes. I mean, are all epithelial cells professional phagocytes? Or like in your kidney, in your breast, in your spleen? You keep calling them professional phagocytes. Is that really, or are these like not quite differentiated fully into terminally differentiated epithelia?
No, I think that they are really terminally differentiated, but to call something like, or some cell professional, it needs, I think that it was originally linked to the efficiency, I think, in clearance, or to the, that they are in charge of the main charge of the cell. So
is this an intrinsic property of all epithelial cells? If you say they're terminally differentiated, they all should do it.
I am not 100% sure. What Cody Chandran says is that they are specialized epithelial cells like the testis or the retina, which are more efficient in clearance, and they are not clearing necessarily apoptotic cells. But what is usually said, and is not clear in my opinion, that is really going on, is that the normal turnover of apoptotic cells in the body, not when you have a damage, but the normal turnover, is performed by neighboring cells, and by neighboring cells you can include epithelial cells, but this was never really proved. So it can be that all epithelial cells are very efficient in clearance or not. I think that we still don't know. What I think that is clear is that all epithelial cells can phagocyte, and you put different cell type of cells, epithelial cells,
can
culture
you
cell
do
lines,
it
and they can phagocyte. But
I'm
if
just
they
curious.
do it efficiently is what is not clear.
Can you do it? Can you take a layer of MDCK cells and they'll do this in vitro?
Yes.
Yes?
Yes, these cells are able to phagocyte. Yes, the several epithelial cell lines are able to phagocyte.
Okay,
They do it with different
question.
efficiencies, with different properties. They don't eat the same, all of them, but they have the machinery for phagocytosis. This is true.
So the second question is, what's the PS receptor and how is it talking to the actin polymerization machinery? And do you think it's mechanosensitive too? That's another question.
Yes. So we still don't have the exact molecular target. We have some clues, okay? And as you can be particularly interested, it can be some integrin working there as a receptor, but we have other candidates and we still didn't identify the proper molecule, okay? But we think that an integrin can be involved.
Cool. That's really just lovely story. Thank you so much.
Thank you.
Such a wonderful talk, Esteban. Our next question is from Ernest Liu says, very interesting talk. How do you know that it's actin pushing the apoptotic cell rather than the apoptotic cell dragging the actin protrusions? And does the apoptotic cell have any migration ability?
No, the apoptotic cells do not have, as much as we know, they don't have intrinsic motility. If we just let them there, we put it in a context in which we have only other stem cells, but we don't have epithelial cells that can push them. They don't move, okay? They are just floating passively, and they don't have intrinsic motility in all the analysis that we analyze, okay? And when we block, for example, in apoptotic cells, all the pattern can control cytoskeletal motility. The motility is not affected at all. So we are pretty sure that the motility of apoptotic cells is generated by other cells, which is something that can be interesting also for when you see in a live tissue things moving. They are not necessarily moving by themselves. No, there's not always, oh, I have these cells that are motile. Maybe they are being moved. No, this is what I think. Yes.
Fascinating. We have a question from Pablo Vicente Munera saying, nice talk. And he's wondering how the volume of the apoptotic cells affects the digestion. Is it the same to digest 100 small cells or one big cell?
Well, I only can speculate about this. So what we quantify is both things in terms of saturation of the epithelial cell. We quantify the number of particles and we quantify the volume. And we observe the same type of behavior. We can either take one criteria or the other, but there is a saturation that, in my opinion, as far as we know, it can be independent. If we ingest a large particle, you are kind of saturated, okay? And if you ingest several small particles, you can also saturate doing this. But this is not something that I was comparing systematically, but I know that there is a saturation from the volume and from the number of particles.
Amazing. Next question is from Niletaneja asking, any thoughts on what regulates the maximal length of the actin protrusions? And then there is a second part, which I'll tell you later.
The maximal length, no. The maximal length, no, I don't know. What I know, there are some molecular players, I mean, actin nucleators that are operating in one or another protrusion. ARP23 is part of both protrusions, while formins are mainly involved in phagocytosis, and apparently they do not play a role in the arms.
Interesting. The second part of Nilay's question is there mechanical disengagement when apoptotic cells are handed over to the next epithelial cell?
How the apoptotic cell is, I understand that is how the apoptotic cell is not, I mean, yes, it's disengaging from the arm. Yes, I don't know. I think that in this case, we have to think in the arms similar to the actin comets, in terms of that they are these actin polymerizations that by polymerizing actin are pushing. And this is driven in that case of the actin comets that are induced by bacteria, is by specific protein. In this case can be also induced by phosphatidylserine, for example, which we saw that is enough to induce this behavior. How exactly this is if we need some addition there in this region or not, is something that is not clear. Probably we have some level of addition, but to some extent, this is important or not is not clear. Yeah.
Awesome. I actually had a question and maybe I missed it. Do you think there are sort of two population of epithelial cells, the one which are pushing and the one which is sort of phagocytosing?
No, it's an interesting question, but it's not the case because you can see both protrusions generated even in the same cell in the same moment. They can generate the two protrusions together, the two protrusions independently. They are not preferential regions of the embryo in which one or another protrusion is generated. As we see, it's not something like the epithelial cell is intelligent and will push when it's saturated. It's just a stochastic process that favors that the targets are spread, and this in general always will improve the efficiency because you are avoiding saturation.
Wonderful.
Great. We have a question from Natalie Hussein saying, great talk. Do these epithelial cells share any markers with the immune cells? Since you mentioned that no specialized cells were present at the time of development that are able to eliminate the apoptotic cells.
Yes, it's something that I didn't show, yes, but we performed transcriptomics on these cells, and they not only share some features with, these cells, epithelial cells of the zebrafish, they not only share features with the trophoblasts of mammals, even humans, but they also share some proteins with macrophages. Okay, so they share molecules with immune cells, and we still don't know how much they can do in addition to phagocytosis, but yes, they have some markers. But this is true. It's true that these markers are present, most widely present than what we thought, no? Because it's like if there are many immune proteins that are not only in immune cells, which doesn't mean that these cells can perform all immune tasks, but they are present and maybe they play other roles too. No? This is true.
Awesome, thank you. I think next question is from Jonas Hartman asking, do you see fusion of phagosomes within epithelial cells? And this could be a hallmark of professionalism as it was shown to increase phagocytic efficiency in microglia and macrophages.
Yes, I didn't quantify this, but I saw it. Yes. They were events for sure. I saw this event of fusion, but we didn't quantify properly this. Yes.
Okay, thank you. Sorry, the questions keep coming in. Are you okay to keep asking as people keep answering as people asking?
Yes,
okay.
It's okay.
Okay, from David Moreno Martinez, thank you for an amazing talk. Is there a mechanism regulating the route of the particle, or is it random? Do the epithelial cells signal to each other to coordinate which one takes the apoptotic cell next? I'm picturing like soccer.
I think that there is a lot of communication in terms of like, all the protrusions that we observed are generated only upon contact between an epithelial cell and an apoptotic cell, but I observed some features that suggest that there are secreted factors from the apoptotic cells that can be activating the epithelial cells. Communication between epithelial cells, there are some mechanical communication. It can be also biochemical, but I still don't know. I don't have any clue about this. About a coordination between pushing and phagocytosis, this is something that I insist. I don't think that is an intelligent machine. I think it's more like a stochastic that they spread and uptake simultaneously.
Awesome. Next question is from Si Chang asking, instead of apoptotic cells, if you introduce other ectopic cells, like for example, dissociated live human cells, do you see phagocytosis as well?
Yes. In fact, some of the experiments that we did for the saturation were done with shortcut cells, human T cells. So they ingest the shortcut to cells, always that they are apoptotic. If they are not apoptotic, they do not detect it. This is a conserved feature that they don't ingest healthy cells, even if it's interspecific. No?
Awesome, thank you. I think that is our questions from the chat. I will say that this blew my mind. I think you broke my brain today. I did not know that this was possible. So just to confirm, these arms come from the basal side of the epithelial cells, yes?
Yes, the polarity of the embryo is kind of inverted with most of the adult organs, in which the lumen is apical, is the apical side of the cells. In this case, the interior of the embryo, which a cavity is, where the cavity is, this is the basal side of the cells, and all the protrusions are in the basal side, in the interior of the embryo.
Okay, and so if you were to put some, apoptotic cells, like if you could get them on the apical side, do you think you would see these, or is it, depending on sort of the specialized basal machinery?
I don't think that they can recognize apoptotic cells from the exterior, and I have some evidence because when the cells that are dying are the epithelial cells, and they can be extruded by the neighboring cells to in the direction of the interior or the exterior. When they are extruded to the interior, they are ingested, but when they are extruded to the exterior, they are not ingested. So I don't think that they can detect. However, there are some people who did some curious experiment with mammal embryos, and they added some particles from the exterior, and they say that if you leave it for a long time, they ended in the interior, but I am not sure how they did it. In my hands, they are not ingesting like macrophages that they can eat particles of metals or something like this. This is not the case.
It's fascinating. All right, we're going over time. So I have lots more questions, but I'm going to go read your paper today because this is really just absolutely fascinating. So thank you both so much. Thank you, Emily. Really fantastic talk, and thank you, Esteban. Really great, and we appreciate your time. And I will sign off from